Hello everyone. So the next topic which we will be discussing is about the differentiation, de-differentiation and redifferentiation. So we will be discussing each of these processes in the plants in detail, and the main focus of the difference will be in de-differentiation and redifferentiation in the following slides. So this is a very short topic which we will be covering today. So let's get started with the first slide of differentiation. That what is differentiation, what is the definition of differentiation, and what is the example of de-differentiation. So uh, the cells which is derived from the root apical and shoot apical meristems and cambium differentiate. So the cells that are derived from the root apical and the shoot apical meristems and the cambium, they differentiate and they perform various specific functions. So this maturation stage is known as the differentiation. So this is the proper definition of differentiation. During the differentiation, cells undergo few to major structural changes both in the cell wall and the protoplasm. So in case of maturation also, there will be a difference in the cell size, and it will lead to the changes in the cell wall and the protoplasm. So here also differentiation is the major structural changes. If we take an example of differentiation, then it will be when there is a formation of tracky elements, the cells would lose their protoplasm. So this is an example. So what they develop is very strong elastic lingocellulic secondary cell wall, from which they carry water to long distances even under the extreme tension. So this is the example of the differentiation, and this is the definition of differentiation. And there are major structural changes which takes place in the case of differentiation. Next we will be discussing about the de-differentiation process. So after the differentiation there will be de-differentiation. So this is a very interesting phenomena. The plants in this category shows a very interesting phenomenon, that is the living differentiated cells lost their capacity and they divide to regain the capacity of division under certain conditions. So in the next slides basically we will be there's a diagrammatic representation in which we will be discussing in detail that what is the differentiation, de-differentiation and redifferentiation. So in this is an interesting phenomenon, and the plants living cells, they lose their capacity to divide and regain the capacity of division. So the fully differentiated parenchyma cells undergo de-differentiation, which leads to the formation of cork cambium and interfascicular cambium. A de-differentiation tissue has the ability to act as a meristem that could give rise to a different set of cells. So the de-differentiation tissues only have the capacity to act as a meristem. The ability of those cells for further differentiation depends on different parameters. So there are different parameters. In differentiation, there were structural changes in the cell wall and protoplasm. In case of de-differentiation, there are different parameters on which the differentiation depends, that are genetic and epigenetic variation. So this concept is used in the plant tissue culture to form a callus. So plant tissue culture, in the case of plant tissue culture, there will be a formation of callus. Callus is basically a group of cells which is being formed in the PTC labs. So this is the concept of used in the practical. If we see in the practical life, it will be used in the PTC to develop the callus, and after this they will form a plant from this. So this was all about the de-differentiation process. After this there will be a redifferentiation. What is redifferentiation? De-differentiation plant once again loses its capacity to divide and becomes mature. In this case it will become mature. It will not regain the capacity to divide again. So it will become mature. So this phenomenon is called the redifferentiation. So what are the examples of redifferentiation? There is a secondary xylem and secondary phloem, which best describe this process of redifferentiation. Then de-differentiated vascular cambium divides further to give rise to secondary xylem in the inside of and secondary phloem on the outside. So So both of them are the best examples to define the redifferentiation. This is how they are showing that dedifferentiation and redifferentiation process are somewhat interrelated. First, there will be a dedifferentiation vascular cambium, which give rise to the secondary xylem inside and the secondary phloem outside. So the secondary phloem and the secondary xylem, they lose their ability for further division. So in this case, they will lose their ability for further division, because secondary phloem and secondary xylem, they are in the mature states. They become mature. After this, there will be no redifferentiation in this. So instead, they become mature to fulfill specific functions of the plant body, which includes the transportation of the food and water and respectively. So these are the mature parts of the plants which will perform the specific functions that will include the transportation of the food and water respectively. So this was all about the redifferentiation process. So next is the diagrammatic representation, in which we will be showing that how this process works. So this diagram is a clear understanding of how this is being worked. So this is a plant cell. The plant cell, there is an external and internal signals which is being given to the plant here. So this will lead to the process of differentiation. So this the two arrows here is that they can be interconvertible process basically. This will lead to differentiation. There is a stem cell-like state, that is a transient state. This process undergoes de-differentiation, and there is a chromatin decondensation here. So this process goes to the de-differentiation state. So redifferentiation and dedifferentiation can be interchangeable. For example, it can lead to dedifferentiation, and redifferentiation can also lead to dedifferentiation. So there are three processes in which it acts dedifferentiation. There will be cell death. Either the cell will die, or there will be multiple copies of the cell. There will be a cell cycle which will come into the picture, or there will be a redifferentiation process or transdifferentiation process which occurs in the plant cell. So this will make us more clear, and this will be in a more understandable way that what are the processes, how it goes, and how differentiation can be converted into dedifferentiation. Dedifferentiation can further be converted into differentiation. Then differentiation process gets converted into redifferentiation, and redifferentiation process gets converted into dedifferentiation. So this was all about the process of differentiation, dedifferentiation and redifferentiation. So in the next slide, we will be discussing the difference between dedifferentiation and the redifferentiation. So this is the differentiation and redifferentiation slide. So in this, we will be first we will understand what is the definition of both the terms, what are the outcomes, what are the new cells which are being formed, and what are the examples of this. Although we have covered it in a separate slide, but to get it in a more clear way, this is the table which I am showing here. So in dedifferentiation, what is the standard definition? The mature cells reverse their state of differentiation and acquire the pluripotentability. But in case of redifferentiation, the process where dedifferentiated cells lose their power of division and become specialized to perform a function by converting into a part of the permanent tissue. So redifferentiation, for example, secondary xylem and secondary phloem, I have given already the examples. These are the mature states of the plant. So redifferentiation lose to the power of division and they become specialized to perform a specific function by converting into a permanent tissue or a mature part of the plant. Then what are the outcomes of both the differentiation and redifferentiation? Cells, they regain the capacity of further division by dedifferentiation, and they have the capacity for further differentiation if lost in the new cells due to redifferentiation. As in the diagram, there was an arrow that is showing it can be converted into differentiation, redifferentiation. So this is about the outcome which is I am showing here. Then new cells, the new cells are formed by dedifferentiation. They act as meristem for further differentiation. But in case of redifferentiation, the cells give rise to secondary structures which carry out specific essential functions. For example, secondary xylem and secondary phloem, they help in the transportation of food and water. So they will perform a specific essential function. Then examples here are the cork cambium and interfascicular cambium. They are the examples of dedifferentiated tissues. and secondary xylem and secondary phloem are the examples of the redifferentiated tissues. So this was all about the differentiation, redifferentiation and dedifferentiation processes. So these are all the references. Thank you.